All right. Okay, so looks like I maybe didn't finish the announcements here. All right. So what you've got on your plate at the moment is that research project write-up, where the part one is you're discussing the current practices, technology that relate to the topic you've selected for your research project. And so that's 3 written pages. Be sure to look at the assignment description because that tells you the line spacing you should use, the font size, and so on. So there's no loopholes in getting, writing less. I tell you specifically how it should be formatted. That needs to be turned in before class on Monday. And then Wednesday, we're going to the water treatment plant. We'll just do the same thing as before. We'll meet in the lobby at 1, maybe a few minutes before 1 if you can. And we'll follow the same groups, assuming that the same drivers are available. If not, let me know and we can make adjustments. We'll just do the same procedure, but this time instead of going downriver to the wastewater treatment plant, we're going to go upriver to the drinking water treatment plant. And it's good that the drinking water treatment plant's upriver of the wastewater treatment plant. That's what you want, and that's where it is. So I'll give you the address and instructions, and we'll talk more about that, but that's Wednesday of next week. And then also Wednesday of next week, homework 11 is due. So we're going to go over a couple of examples today in class that I think are going to help you with homework 11. So any questions related to the announcements? All right. So we're going to actually focus on activated sludge exclusively today. We've finished up primary treatment. So activated sludge, remember, is where we are recirculating sludge to try and increase the amount of microorganisms to consume the food. The food that we're wanting them to break down is just anything, any kind of biochemical oxygen demand that would cause the dissolved oxygen to decrease if it was discharged into the river. So the simplest kind of secondary treatment is, what if we had a reactor where we didn't recirculate the sludge? It would have to be really big. If there was no recycling of sludge solids, and if we just had complete mixing and we provided aeration, then the mean cell residence time would be really large. And in the case of no recirculation, then the hydraulic detention time and the mean cell residence time are the same. Mean cell residence time is talking about the average microorganism, how long it spends getting recirculated through the aeration basin and the secondary clarifier. So in a minute, mean cell residence time will mean more because we'll actually be recycling sludge solids. Now, in this no recycle, it's just a big tank where the water goes and a little bit of the BOD would be broken down depending on how long we leave it in there. So no recycle is really only used in big rural, large treatment systems that serve rural areas, and they're large because they're big in land area, because they have to be if there's no recirculation of sludge solids. So the way that you'd calculate the mean cell residence time here is it's just the same as the hydraulic detention time, which is the volume divided by the flow rate. So this slide's not important, really. We won't spend too much time on it. It's not important because it's not really what's going on at most treatment plants. At most treatment plants, they're going to recirculate the sludge. And that's because the reason why this isn't done very often is because it gives you poor control over the number of microbes that are in the aeration basin. If you're not recirculating the sludge solids, then the number of microbes that are in there just are a result of the temperature and the wastewater characteristics. We don't have anything that we can do to adjust that. And because of that, things are happening very slowly. And that means that a large tank volume is required. So because of that, it's rarely used as a main method for breaking down the BOD. What's far more common is it just to be an aerated lagoon and occasionally used in rural areas where land isn't expensive. maybe you might as well just have a really big aeration basin, and that would be simpler, require less like human interaction than constantly tinkering with the sludge recirculation rate. This is a diagram of what's far more common. And this figure maybe looks a little bit complicated at first, but it's not so bad. So this is the stream of wastewater that's coming in from the primary clarifier. So it's coming in from the top of the primary clarifier. Remember, the bottom of the primary clarifier is sludge, which is either going to a digester or some sort of dewatering system, whether it's dissolved air flotation or a belt press that's squeezing the water out, or sometimes they just take sludge and they land apply it and let it dry out, like sludge drying beds. Whatever the case may be, the flow that's coming into this system is the water from the top of the primary clarifier. So it's got BOD, it's got suspended solids, and we want to break down the BOD in the aeration tank. So it's got some flow rate and some S0. That's the initial concentration of substrate. That's the BOD that's flowing in. The aeration tank, the parameters that we care about inside the aeration tank are its volume, the concentration of substrate in the aeration basin, and then X, where X is the concentration of microorganisms. So when we recirculate sludge, we can increase the concentration of microorganisms. And so we can mean that there's a lot of bacteria in there, a lot of hungry bacteria breaking down the BOD much faster than if we were just relying on the amount of bacteria that are normally present in wastewater. So we're like inoculating the aeration basin with the return sludge. So there's actually two streams coming into the aeration tank. There's the water stream from the primary clarifier, and then the sludge stream. So we're re-injecting sludge into the aeration basin so that we have control over X. That's the main reason we recirculate the sludge, is so that we can control the concentration of microorganisms. So then what comes out of the aeration basin, you'll notice here that the flow rate of what's coming out of the aeration basin was the liquid flow rate Q plus QR, which is the volumetric flow rate of the return sludge. So it's the combined flow rate comes out, and then it has the same microorganism concentration and substrate concentration that's coming out. Because it's well mixed, the concentration everywhere in the aeration basin is the same as the effluent concentration. But that's not the same as the inflowing substrate concentration. So S0 is much larger than S. So the water that comes out of the aeration basin is settling in the secondary clarifier, and then the sludge that goes to the bottom, a portion of it is returned and a portion of the sludge is wasted. So this is the waste sludge. And so X is the concentration of microorganisms. Here where it says R, that means in the recycle. So off the bottom of the secondary clarifier. So then what comes out of the top of the secondary clarifier is the water component, which hopefully has very low microorganisms. We don't want X, the concentration of microorganisms, to be high in the effluent of the secondary clarifier. We also don't want S, the BOD concentration, to be very high. So this is a schematic that I think you're going to have to look at this and think about it. Anybody know what these dashed lines represent? These dashed lines are telling you to do a mass balance. It means consider the flow in and the flow out. There's going to be balance in the volumetric flow rate. So Q in equals Q out. Now, the S, the concentration of substrate out, should be lower than the concentration that went in. So what happens is the microorganisms, they are reaching into the water and taking food, which is like dissolved BOD, and they're turning it into cellular mass. they're turning it into like their body mass. And so that's what gets settled at the bottom of the clarifier. So this X that's getting wasted includes like the broken down components of BOD that otherwise would have been discharged to the river, because the microorganisms are turning, basically you can think of it as sugar water, into body weight. And it's not actually sugar, it's any kind of food source, whether it's carbohydrate, fat, protein. They're taking the BOD, turning it into body mass, which can be settled. We couldn't settle it before they turned it into microorganisms. That's why what we want them to take out the BOD is because we can settle microorganisms, but we can't settle dissolved BOD because it's dissolved. So any questions about this figure, which is really important? We have an exam coming up next Friday, right? Be good to be able to explain this figure. It's pretty important. All right, so let's talk about the design of secondary treatment. Some of the things we care about is how big the reactor is. We care about the recirculation rate of sludge. So how much of the sludge we send back into the aeration basin versus how much of the sludge we discharge for disposal. And by changing the recirculation rate of sludge, we have control over the food to microorganisms ratio. So we want there to be more microorganisms in the aeration basin than there otherwise would be naturally, and we accomplish that by recirculating sludge. We also care about how much air we are putting into the aeration basin, and so like a typical rule of thumb would be that for every cubic meter of wastewater that you're treating, that you're going to need 8 cubic meters of air to be blown into the aeration basin to ensure aerobic conditions and make sure that the microorganisms have the oxygen that they need to break down the BOD. Because the microorganisms are consuming two things. They're consuming BOD, but they're also consuming oxygen. So we have to provide both. It's like we're throwing a party in the aeration basin. And the guests are the microorganisms. The food is the BOD that comes in, but then they also need air to breathe in that party that we're assembling in the aeration basin. The secondary clarifier, we care about how well it's able to settle mixed liquor volatile suspended solids. This abbreviation here, MLVSS, that means volatile suspended solids is a surrogate for the bacterial load. So how many microorganisms? Suspended solids just means anything that's not dissolved. So it means like solids that are suspended in the water. Volatile suspended solids is saying, well, let's exclude the inorganic component, meaning like salts or anything else that's inorganic. So the volatile suspended solids is bacteria. And then mixed liquor, which is a really strange name for it, but mixed liquor is what we call the stuff that's in the aeration basin. So when we add wastewater and return sludge, that's mixed liquor. So that's our design parameters. And then once it's built, the operators, one of the biggest levers that they have to pull on to like change the performance once the system's already been built, they can change how long the cells, how long the volatile suspended solids spend in the system by adjusting how much sludge is wasted versus how much is recirculated. So you remember in this figure there's a split. So, you know, is it 50-50? Do you want half the sludge to be recirculated and half the sludge wasted? The valve that directs a portion of the sludge to recirculation and a portion of it to waste, that is how you control the F to M ratio, the ratio of food to microorganisms. F, the food, is just, that's a reflection of the conditions of your system. You don't have direct control over that, but you can control the number of microorganisms in the aeration basin. So they know like a target mixed liquor volatile suspended solids concentration. And I think when we visited in Huntington, I believe he mentioned that they were aiming for about 2,500 milligrams per liter as their F to M, as the mixed liquor volatile suspended solids concentration. So F to M ratio. The way that you calculate that is here as shown. It's the food is Q times the substrate concentration that's coming in. So that gives you how many milligrams of BOD per day are coming into the treatment plant. That's the food. It's a mass flow rate of food. And then the denominator in microorganisms is the volume of the aeration basin multiplied by the concentration of mixed liquor volatile suspended solids. And so that gives you the mass of mixed liquor volatile suspended solids. If you've got the concentration multiplied by the volume, it gives you the mass. So food to microorganisms ratio is calculated using this formula. So the more you waste the sludge, the lower the concentration of mixed liquor volatile suspended solids will be. Like if you wasted all of the sludge, then the mixed liquor volatile suspended solids would only be the same as what's coming in. But if you recirculate most of the sludge, then your mixed liquor volatile suspended solids will be even higher. So if you have a high sludge waste rate, that means you're going to have a low mean cell residence time and a high ratio of food to microorganisms. So if microorganisms is relatively low, if you don't have many microorganisms, then the ratio of F to M is high. Now, if you have a high F to M ratio, then that means that the microorganisms are saturated with food. They may not be breaking down the substrate as completely. One of the advantages of having a relatively low F to M ratio is that it means that the microorganisms are more starved, and they kind of, there can be more variety of the biological, like more biological variety in the types of microorganisms that are there. You can have some predators that are breaking down simpler producer type microorganisms, and the waste is degraded more completely. You'll have less sludge volume if you have a low F to M ratio. So a typical range of food to microorganisms is between 0.1 to 1. So think about what that means in physical terms. That's like what we're saying is the bacteria are being offered between 10% and 100% of their body weight in food each day. That's the typical operating range of F to M ratio. So 0.1 means 0.1 milligrams of food per milligram of microbes per day. So microbes can eat more than we can. Now, I'm about 160 pounds. I can't eat 160 pounds of food per day. But the microorganisms, potentially, that's what we offer them, is up to 100% of their body weight per day in food. So here are the design equations that we use and some typical growth parameters. We're going to do an example here in a minute. So let me write down some of these typical growth parameters on the board because we're going to need them later. So hydraulic detention time, we've seen that plenty of times before. It's just volume divided by flow rate. Mean cell residence time is how long does a bacterial cell spend on average circulating around between the clarifier and the aeration basin before its turn comes up to be discharged. So like think about from a probability standpoint, a cell, when it gets to the valve that's a portion of its recycle, a portion of its waste, you know, statistically speaking, some of the time it's going to be recycled, but eventually it will be its turn to discharge. And what theta sub c calculates is how long in time it spends in both the aeration basin and being recirculated between the aeration basin and the clarifier before being discharged. This formula tells us the concentration of BOD in the effluent as a function of some kinetic parameters like K sub s, which is just a lab test you do to find out what's the concentration of BOD that's associated with half the maximum growth rate for your population of bacteria? So like there is a certain optimal concentration that maximizes the growth rate, but like for half the maximum growth rate, then what's the BOD concentration that allows your population to grow at half that rate? That's K sub S. That's the physical explanation of it. Then the other parameters are related to how quickly the microorganisms grow. So this mu sub M is the maximum growth rate, and then K sub D is related to the death rate, or the decay rate of microorganisms. And then finally Y, the yield coefficient, is basically how much weight the microorganisms gain for the food that they eat. So if I eat a pound of food a day and I gain half a pound, then that means the yield was 50%. So the typical yield, you can see, goes between 0.4 and 0.8. So they don't turn 100% of the food that they eat into additional body weight. The difference being that there's some amount of the energy that they're consuming which is used for cellular respiration. It's just the energy they consume for metabolism, and then some of the energy that they consume goes into new cells that are growing. Because remember, they're going through the process of binary fission. They're dividing from one microorganism to two, 2 to 4, 4 to 8. And that binary fission leads to additional mass, and so the yield accounts for that. So what I'm going to write down is K sub S is 60 milligrams per liter, because we're going to use this in the example for a minute. K sub D is 0.06 inverse days to the minus 1. Mu sub M is 5 inverse days. And then yield, the typical yield, would be 0.6, and that's unitless because it's the ratio of volatile suspended solids, which is their body weight, to the ratio of BOD, which is their food. So yield is just how much body weight they add based on how much food they're eating. Anybody have any questions so far about these growth constants? So this formula allows us to understand what will be the concentration of BOD in the aeration basin and in the effluent. And the interesting thing is it doesn't depend on the concentration that's coming into the aeration basin. Not directly. It depends on other things like the kinetics, and then theta sub C is how long the cells spend in the aeration basin. And so where we do account for the incoming concentration of BOD is here when we're calculating X, which is the concentration of microorganisms in the aeration tank. So this is saying that the difference between the incoming and the outgoing concentration is going to tell you what concentration you need to have in the aeration tank. So, if you know what your incoming concentration of BOD is, and you know what the outgoing concentration of the BOD needs to be because of your permit, then that would help you to calculate the desired mean cell residence time that ought to go into this formula. So I'll show you in just a minute how this all fits together, but are there any questions so far? Let's look at an example. Let's say that we had a wastewater treatment plant where 0.25 cubic meters per second is going through the treatment plant. And their standard, which is set like by a regulatory agency, is that they have to achieve 30 milligrams per liter of BOD and no more than 25 milligrams per liter in suspended solids. And the thing about the suspended solids is that a portion of those suspended solids will be inorganic, but then the suspended solids also contribute to the BOD. So we have to take that into account when we find out what's the allowable dissolved BOD concentration. So we'll account for that when we go through this example. This is saying that the BOD in the primary clarifier is 115 milligrams per liter. So that's what comes into our aeration basin, because here's the primary clarifier, and then it's our aeration basin with lots of little bubbles in there, the secondary clarifier. So this is primary sludge. And here's the effluent from our primary clarifier, which is going to go in here. So this example is saying that the effluent of the primary clarifier is 115 milligrams per liter BOD. So S naught is 115 milligrams per liter that's coming into the aeration basin. And then it also has suspended solids. Suspended solids is 65% BOD. And the mixed liquors of volatile suspended solids that we're going to go for is 2,500 milligrams per liter. All right. So first of all, on this question of what's the allowable S, the water that comes out of the secondary clarifier has two things in it. It's got BOD and suspended solids. The suspended, by BOD, what I mean is dissolved BOD. So there's dissolved BOD, and a portion of the suspended solids that come out are also BOD. So it's like floating particles of organic material that exert biochemical oxygen demand. So the allowable BOD is going to be reduced because our permit says 30 milligrams per liter are allowed. So let me write it over here on this board. S is going to be the BOD 5, which is allowed, minus the BOD 5 in suspended solids. Okay, so the amount that's allowed is 30 milligrams per liter. The amount that's in suspended solids is going to be 65% of our limit. Our limit is 25 milligrams per liter. 25 milligrams per liter. So that means that we can only have 13.75 milligrams per liter of BOD 5 in solution. So the BOD that comes out of secondary treatment, some of it is dissolved and some of it is suspended. The suspended part of the BOD will be 65% of 25 milligrams per liter. And then the dissolved fraction of BOD will be 13.75 milligrams per liter. So let me bring up this example for the benefit of people who aren't here today. I heard, by the way, that the Marshall team won the environmental contest at the Virginia's symposium, so that's good. Okay, so what we did so far is this, that S, we're trying to find out what concentration of substrate dissolved is allowed in the aeration basin and in the effluent. 13.75 milligrams per liter, because we have to subtract out the contribution of suspended solids that are BOD. Okay, so we did that. Next, find what is theta sub C. So here's the equation we're using, but the unknown is theta sub C. So let me write it over here on the other whiteboard. So S is equal to K sub S, 1 plus K sub D times theta sub C, divided by theta sub C times U sub M minus K sub D minus 1. All right, so we know that it's 13.75 milligrams per liter is what's allowed. And then we know the kinetic coefficient is like 60 milligrams per liter for the case of S. 1 plus case of D is 0.06. And then that's multiplied by the theta sub C. And then in the denominator, it is theta sub C again, multiplied by mu sub M. That's 5 minus 0.06, and then minus 1. So this one's a little bit tricky because the unknown that we're trying to solve for exists in more than one location, because we've got theta sub C here in the denominator and also theta sub C here. We can just algebraically move things around a little bit. Let me show you what that looks like. 13.75, 60 times the death coefficient in the numerator, mean cell residence time times the difference between the growth and the death constant. So if we multiply that out, we'll get that theta sub C here, the mean cell residence time is 1.146 days. So in this process, that tells us that the average cell should spend a day, a little more than a day, recirculating between the aeration basin and the clarifier before it's discharged. Now, what should be the hydraulic detention time? That's different from the mean cell residence time. The hydraulic detention time is just related to the flow rate and the size of the aeration basin. But to know what hydraulic detention time is required, we have to look at the difference between the incoming and the outgoing substrate concentration, along with things like the mean cell residence time that we just calculated, our intended mixed liquor volatile suspended solids, and so on. So we know X is 2500. We know theta sub C we just calculated is 1.1. In fact, we know everything here except for theta. So here, if we use this formula and we're saying 2500 milligrams per liter for X, and rearrange this equation for theta. So it turns out that the hydraulic detention time should be 0.026 days, which is 2251 seconds. So that's how long the water should spend inside of the aeration basin. And then if we know the flow rate that's coming into the treatment plant, we can then determine the volume of the aeration basin, because it's going to be the flow rate times the detention time. So that's the next thing that it asks, is it says, so we already figured out how long the cells will spend in the tank. We already figured out how long the water should spend in the tank. So how large should the tank be using this formula? So the size of the aeration basin we can determine just by multiplying it by the volumetric flow rate, 0.25 cubic meters per second. So the aeration basin should be 563 cubic meters. And then just the last piece of the puzzle here is finding out the sludge wasting rate. So remember, a certain fraction of the sludge should be recirculated, a certain amount should be wasted. And so to achieve the 2500 milligrams per liter of mixed liquor volatile suspended solids, that is going to be the concentration of microorganisms in the aeration basin. There's more in the sludge. So this is saying, what if the return sludge has 6,000? What does that tell us about the rate that the sludge is being wasted? So that X sub R is the 6,000. If we're assuming that there's 6,000 milligrams per liter of volatile suspended solids in the sludge that's being wasted. So the volumetric flow rate, the rate that the sludge is being disposed of, is 205 cubic meters per day. When we put in the volume of the aeration basin, the concentration of mixed liquor in the aeration basin, the mean cell residence time, and the concentration of mixed liquor volatile suspended solids in the sludge after it's settled in the primary clarifier. So, I mean, I recognize that these formulas are pretty foreign the first time that you see them, but you'll get some practice with these ideas on the homework assignment that's due next week. And this illustration is just meant to show you like what step-by-step procedure you go through. First, and the homework, by the way, is intentionally set up to be very similar to this example. So, you know, this example is recorded. If you didn't get a chance to write everything down, the calculations are included in the video. But what you do as a starting point is you just find out how much dissolved BOD can you have. You know, we know that the total amount of BOD that's allowed to go out to the river is 30, but some of that's going to be in the suspended solids. So the soluble BOD should only be 13.75, and then everything kind of just snowballs from there, where you're calculating how long the cells should spend in circulation before they're discharged, finding out the hydraulic detention time, the reactor volume size, and then the sludge wasting rate. So the homework asks you to go through basically that same procedure. Does anybody have any questions about this activated sludge example? So your book has this figure which is showing what happens as a function of temperature and also how long you keep the solids recirculating. And to achieve a high amount of removal of BOD, you want to be on the outside edge of this curve, in this design range as it's called, where you want to have, you know, either if you have warm water temperature, then that makes everything go faster. If you've got warm temperatures, the bacteria are able to break things down more quickly. So here you can either have some combination of either a long solids retention time or warm temperatures to be in the design range. And then this figure is also showing a certain range where nitrification will occur. And nitrification is the removal of nitrogen, which is typically very stimulating to aquatic plants. It's a nutrient that we want to remove if possible. And so if you have a long mean cell residence time, it has a variety of advantages. It means that you're going to have less waste sludge, you're going to have a lot of BOD being broken down, and the removal of nitrogen is good as well. Sometimes to achieve really good performance in the wastewater treatment plant, they may have to add nutrients depending on, you know, whether there's phosphorus or nitrogen in the wastewater to begin with. Sometimes it's not in the optimal ranges, and so it's kind of strange to think of it, but it's like vitamins for a wastewater treatment plant. In the same way that you and I take vitamins to get like the right balance of, you know, B12, vitamin D, and so on. So besides BOD and air, the other thing that these microorganisms need critically are nitrogen and phosphorus. And if you don't have enough nitrogen or enough phosphorus, it's possible that your aeration basin isn't going to break down all of the BOD that it should. So occasionally nutrient addition will be added, but not all the time. Metals can accumulate in the sludge, and so, you know, that's toxic. So, and that's why we're not able to use the sludge for land application in Huntington. They talked about how the metals are too high to be able to use it, like in people's gardens and as an opportunity for recycling those nutrients. So just to revisit some of the terminology that we've talked about so far, I've already told you about MLSS, mixed liquor suspended solids. A bulking sludge is a sludge that doesn't settle very well. And that's a problem. If we're trying to recirculate microorganisms into the aeration basin, one of the biggest problems you can have is that the sludge doesn't settle well. And if it doesn't settle in the secondary clarifier, then we're not able to pump it and recirculate it. And so that's going to threaten the whole operation if the sludge doesn't settle well. And there's a variety of things that can cause a bulking sludge. For example, if there's algae in the secondary clarifier, those algae are going to be giving off little air bubbles, oxygen bubbles, that cause the sludge to float rather than to settle. Or if the sludge particles are so small that they're settling downward very slowly, then that would cause poor settling as well. We've talked about the F to M ratio and how important that is because it's our way of speeding up the treatment process. So at the two extremes of F to M ratio, at one end of the extreme we call extended aeration when we have a low ratio of food to microorganisms. So that means a lot of microorganisms and we're kind of starving them a little bit. If you've got a low F to M ratio, it means you're not giving very much food to the microorganisms. And to achieve that, you'd have to recirculate the sludge a lot because what you'd be doing is you'd be bringing up the concentration of M. To achieve the low F to M ratio, you boost the M by recirculating a lot of the sludge. So that'd take a long time to break down all the BOD if you're doing it that way. High rate aeration is when you're giving the microorganisms a lot of food, so it's less efficient, but the tanks don't have to be as large because the microorganisms are acting more quickly. They're kind of like kinetically speaking more active since they have plenty of food in high rate aeration. When we settle the sludge, how much is going to be available to recirculate partly depends on how well it settles in the secondary clarifier. We talked about a bulking sludge that doesn't settle well. Sometimes we quantify the settling performance through something called the sludge volume index. It's just a lab test that you can do where you like scoop a graduated cylinder into some sludge and you allow it to settle for 30 minutes and then you would find out in like a 1 liter graduated cylinder how much of it is sludge and how much of it is supernatant or is water. And basically you'd have a layer of water and a layer of sludge. And so then after you did that, after you found out what volume of it is sludge after 30 minutes, then you'd filter out all of the solids and you'd dry them and you'd find out what was the mass of the solids. And so a sludge volume index of between 80 and 150 is typical for mixed liquor volatile suspended solids of 2,000 to 3,500. So this is just like how quickly it settles and how well the sludge compacts is assessed by the sludge volume index. So this is just kind of a visual representation of what it looks like when you take some of the liquid, allow it to settle for a certain amount of time. You find the settled sludge volume, how much of it is supernatant, and it's the sludge volume divided by the mixed liquor volatile suspended solids is how you calculate the sludge volume index. Okay, remember I told you for the exam you ought to memorize each of these numbers and what they're called. So bar rack or screen, grit chamber, primary clarifier, and so on. So we've talked about recirculating sludge. We've talked about the performance of the aeration basin. We've seen the design equations for that. Another piece of the puzzle is how much air is required and how much sludge is being produced. So there are formulas that address both of those things. How much air needs to be provided and how much sludge is produced. So the sludge production depends on how effective the microorganisms are at turning dissolved BOD into cellular mass. So we've talked already about yield. So substrate utilization expressed in terms of yield varies between 40% and 60%. And in addition to that, there's the effect of cell death. So it's an observed yield, takes into account the yield which is the, for any one generation of bacteria, the ratio between the food it consumes and the additional mass that it puts on. But then the observed yield also takes into account the rate of cell death that's occurring inside of your system. And so if you know how long the microorganisms are spending in circulation, theta sub c, and you know the kinetic coefficient related to cell death, then this observed yield kind of takes both things into account. And so the observed yield is going to always be the lower than the yield, because you're dividing by some number that's greater than 1. So the observed yield is actually less because you have a fraction of your microorganisms that are dying in any given day. And why that's important is that the sludge production, P sub x, tells you the mass of volatile suspended solids per day that's being generated by your system. And this is, by the way, the mass of sludge solids. It's not the mass of liquid and solids. This is the mass of cell solids, P sub x, that's being generated each day. Okay, 148. What I'm going to do is I'm going to email you the solution to this in-class exercise. I guess I might as well give you the paper since I've got it here. I want you to have an illustration of how you can calculate the amount of sludge that's being generated. The first time I learned about this was in 1999. So, and I've studied it, like I've been through this course and other wastewater related courses probably like 15 times. So here's the point I want to make. Is like, I feel like I have a pretty good grasp of this stuff. I'm not an expert. Like, I'm not varsity when it comes to activated sludge. Like, I understand it. But I studied a lot of times to understand it. You want to be able to do the assignments and be prepared for the exam. Just listening to what I told you today is not enough. 100%, it's not going to get you where you need to be. You absolutely have to read the book related to this to be ready for the exam. This is, activated sludge is probably the most abstract thing that we'll do all semester long. Like there's more moving parts, and just conceptually it's a little bit confusing because you're recirculating streams. So I just really want to emphasize the value that you'd get from investing an hour or two reading through the book and just thinking about activated sludge and what's happening. Because I mean, it took me at least a long time to really understand what is mixed liquor volatile suspended solids, why are we recirculating the sludge, and then how all these equations fit together. So we're out of time. I'm going to email you the solution to in-class exercise 28. Look for that just within the next few minutes. And if there are questions, we can talk more about it on Monday.